Right, so I think, okay, I am recording this now. So I shared, so with the book that we're using, and I think I typed this, but I'm not sure that I mentioned it verbally. Okay, the book that we're using, I have it as PDF, but I also just found a link to it on the internet. So I've been posting the links because I think it's more updated, but the chapter numbers don't match with the chapter titles. So go with what the title says. So if it's facilities, look at facilities. It might say chapter 3 or chapter 4 or something. They might not match exactly. Okay, so the objectives of this is it's going to start to introduce the framework about how the facilities are designed. That's really kind of intense because of the way that the um, the way the products are going to be used, right? So they're going to be injected into someone's body. So when that happens, what we're trying to do is make sure that it's as sterile as possible. And the FDA has very, very strict regulations about that. So they design the facilities kind of to do what they're supposed to do. It's different, right? It's not like they have a building and then they set it up to do the job. They design the whole building in order to do this process. Okay, so we'll talk about all the different parts. We'll talk about how things flow through the facility, the room classifications, basic controls, and a little tiny bit about the instrumentation and control systems. Okay, and I hardly talk about the utility part either. Okay, so one big thing to remember is most of these places, they have lots of engineers that work there. There are biologists that work there, but it's pretty intense how the facility actually has to work. So there are lots of bioprocessing engineers that do that job. Okay, so biomanufacturing facilities are similar to any other manufacturing site, like a car or, let's see, what I'm trying to think. Usually we use car manufacturing to compare it. So any place where somebody's making something most of those places have the same sort of setup. Okay, so they have the place where the manufacturing is going to occur. They have the warehouse where they're going to store the materials, the raw materials, the shipping and the receiving. And then they're going to actually have controls to make sure that the materials that they're receiving are going to do the job that they need to do, that they're approved and they meet the quality that they need to have. There's going to be maintenance and custodial areas. So cleaning is really important, making sure that everything is working properly. HVAC, so the heating and the air conditioning are super important, especially the air conditioning, because all these processes generate a lot of heat. Utilities, so does it have water? Does it have steam? How is the water of a certain standard? Okay, it has to have information technology. Security is key. We don't want to have anything be messed with, tampered with. Office space, locker rooms, break rooms, okay? And there's way, way more stuff, okay? But that's just the beginning part of it. Okay, so the primary element when they design facilities to make these biologic products is to protect the product. Okay, we don't want it to be tampered with or have any adulteration. So we don't want anything to be contaminated. So the way that we protect the product is to ensure that the process is correct and it's protected. Okay, so these facilities are different, right? They're not the same at all. So it's not one size fits all. It's not like they make a big square building or rectangle building and just put the stuff in it. it's really specific how it's designed. So it's going to be based on what product and process they're going to make, how complicated is the process, and are they going to try to make one product there, or are they going to try to make more than one product? And I know this seems so, to me, it just seems, I guess, a different way of thinking, right? Because in teaching, we have a lab and we can teach whatever in the lab as long as we can put the equipment in there. But these facilities, the way that they're designed and the way that the FDA approves it, is you can only make certain um, products in there, and they don't want any cross contamination. So, say you're making COVID-19 vaccine, okay, and then the pandemic is over, they don't need the vaccine anymore, so you want to try to make flu vaccine. It's not going to be a straight, okay, let's just clean it up and start making flu vaccine. It has to go through a lot of regulatory processes. It, it switching over is not straightforward and in a regulatory aspect, and probably in a I guess a process aspect either. Like, we just met with this man and he's trying to start small production facilities, a whole bunch of them, for drugs that are orphan drugs or things that they don't need a whole bunch of it. And instead of being able to just make one big building and have lots of little like subunits of it, labs within it, he's talking about making like 30 to 40 small labs. So building these facilities all across the state. So it's totally different. It's not, it seems, to me, it seems inefficient, but they don't want, if you're using bacterial cells for one thing, they don't want those to cross contaminate and cause a problem with the animal cells that you're using for something else. Okay, so the regulatory framework is important. Okay, so the FDA is the Food and Drug Administration, right? And they're going to provide the general guidelines. And there's a document that's called the CFR, so the Code of Federal Regulation. And it used to be a big book, now it's on the internet. Okay, and so everything is in this document. Okay, so buildings and facilities used in the manufacture of intermediates and active pharmaceutical ingredients, okay, they call those APIs. So there's another acronym for you. Should be located, designed, and constructed to facilitate cleaning, maintenance, and operation as appropriate to the type and stage of manufacture. Okay, so the whole idea is to minimize contamination. So the process is the product, or the product is the process. So we have learned through many, many years of manufacturing that you can't just test the product at the end. Okay, it's not very efficient and it doesn't catch all the mistakes. So what happens now is all along the way, all of the steps of making the product are tested in order to get to this end. Okay, now other agencies that are involved, so OSHA, so Occupational Safety and Health Administration. So this is going to make sure that the workers are okay. So nothing that you're doing in the facility is harming the workers. The Environmental Protection Agency is making sure that the things that are being released from the facility, so maybe the wastewater, is not going to affect the environment. There's going to be building codes, fire protection, okay, and then the CDC. So the CDC is ensuring that any of the living organisms that you're using in there are not going to get out and cause problems somewhere else. Okay, so let's talk about the 4 levels of biological threats. Okay, so these are the biosafety levels. So lots of times they say BSL. Okay, so level 1 is where level 1. We do some level 2 stuff, okay? So undergraduate, secondary training, and teaching labs. Okay, so the organisms that we work with do not cause disease in healthy adults. Okay, so the way that we control it is that we contain it. So we do like bleaching everything, autoclaving, making sure there's hand washing, safety glasses. Okay, BSL2 or clinical diagnostic and some teaching labs have a little bit more. Okay, they have moderate risk agents, so it would have to get through your skin, through your mucous membrane, or you would have to eat it. Okay, what we're doing is one, we don't work with anything that can infect you. So you were going to use biological safety cabinets. So we do use those. I show you how to use those. Gowning, gloves, and when we centrifuge something, you would put it in these little protective cups so aerosols aren't released. Don't want anything to get out. Okay, so here is a biological safety cabinet, also known as a laminar flow hood. Okay, this is not a fume hood. Okay, it's not the same. So fume hood is exhausting air, so you don't breathe the fumes from something. This is to um, protect the organism that's in here so it doesn't get contaminated. Okay, and we don't want to contaminate the organism. I'm pretty sure this is a cell culture lab because this is the inverted microscope and a carbon dioxide incubator. Okay, now BSL3 are clinical teaching, research, and production labs. So these are things that have a potential for respiratory transmission. So you could inhale it. So these could cause serious or lethal infections, but there are treatments available. Okay, so you're going to control aerosols with it even more. So no flame. BSL4 is extremely dangerous things that could be transmitted with aerosols. So these are things that there's no therapy or vaccine available. You do it in complete isolation. You use positive pressure suits. So this is the kind of thing that you see on TV and movies, right? Or at Fort Teacher or anywhere where they're working with fours. So a positive pressure suit means that the pressure is greater inside the suit. So if you happen to get a hole in it, the air is going to go out rather than things going into it. Okay, so that's going to help protect you. Okay, so the facility design depends on the level of threat. Okay, so if you go to Fort Detrick and you're working in something that's there's going to be showers, there going to be special ventilation systems that are going to clean the exhaust. Access is going to be really controlled. You have to demonstrate that you know what you're doing before they let you in. Oftentimes, like when a student gets upgraded or promoted to be able to go into the higher levels, usually what happens is someone working for the federal government, someone comes and checks on their background. Okay, so it's very controlled access. They don't just let anybody in there to do it. The facility is going to be segregated. So there's going to be certain, it's really secure. You're going to have a swipe card. You're going to, like the BSL4 is not going to be by the break room, that kind of thing. Okay, now, whenever they build, the things that they take into consideration is the materials. Okay, so the walls, the ceilings, and floors have to be impermeable to chemicals and disinfectants. Usually we don't have any wood. Okay, there's no cracks. Okay, so everything is sealed up. Whoops, I can't type. Okay, or I'm not even typing, I can't write. So everything is sealed. Okay, if you look in our lab, it is not designed to these specifications, okay, because we have square tiles. Okay, things could get in between the cracks in the tiles. We have horizontal surfaces where our lights are hanging from the ceiling, so things can get up there. We have wood. I typically don't have wood because it's hard to decontaminate, stuff grows on it. Um, it's slip resistant floors, the sinks are no contact. We're not designed this way. This is not what we're doing. Okay, that all the emergency equipment is going to be easy to find. Okay. All right, so hazardous material facilities. Okay, so the outer and the inner change rooms are going to be, okay, so here's the outer. So you first go in to the facility. Okay, so you first go in, you have on your street clothes. You go in here and you put on scrubs. Okay, and then, well, it depends. Okay, so maybe go in, you're not going to go in. Wait a minute, let me erase this. Okay, I think I have pictures. Okay, so you're not going to go in and out the same door. Okay, so if you're going in, you're going to come in this way. When you come out, you're going to go out that way. So lots of times when you go into work, you're going to put on scrubs. Okay, and then from the scrubs, you're going to go into another room where you're going to put on your sterile, where you gown in. Okay, and then you go in here and you do your work. Okay, and then when you come back out, okay, the outer layer comes off here and goes into certain containers. Then you're going to shower, and then you're going to come out. Okay, sometimes you're going to shower if you're in one of these bunny suits, okay, like this. That's going to be disinfected before you go out. It totally depends on what you're working with and how bad it is. Okay, so you could have a chemical decontamination area for the suits, is what I just tried to explain. You have to also take into consideration how you're going to decontaminate your materials and your supplies. So lots of times the materials and supplies stay in here. Sometimes they'll go through their own little door in an autoclave and they'll be disinfected that way, or they're going to go through a UV light. We had an opportunity when they were building, so down in Frederick, the Frederick National Labs, and I'll see if I can find pictures of this. They were building a facility that was BSL 3 and 4, and they were doing biomanufacturing. So they were building this facility where you had to protect the product and you had to be protected. And we were able to tour it when they were building it, so we could see some of the flow of how this would be. They don't, students will never be able to afford it. It's just not safe. They don't let you do it. Okay, so we had the opportunity to actually see it when they were building it, which was really cool. So nothing was happening in it yet. I'll see if they have any videos about it. I actually haven't heard about it in a long time, so I need to follow up. Okay, so I already talked about the exhaust air ventilation system is going to create negative pressure in the work area. And I tried to explain to you how these positive pressure suits are going to work. They're going to have a life support system, and there's going to be HEPA filtration on it. This is if you're in 3 or 4. Okay, which they're going to train you before they ever let you in there. Okay, now, when you're designing facilities, things that you have to take into consideration are different things like temperature, flow, how do materials flow through the facility, air, and then the characteristics of the product. So is it going to harm the employees? Will it harm the environment? Is it stable? Like, does it have to be at a certain temperature? How big is it? Is the product going to be in these big, huge vats, or are you going to minimize it? So there's just a whole bunch of stuff that goes into it. Okay, now when we talk about the functional areas of the site, they also call them units, right? So how these units are going to operate. Okay, all of this stuff is regulated by CGMPs. Okay, so C stands for current, and GMPs are good manufacturing. Oh, I'm sorry, guys. Manufacturing practices. Okay, so lots of places in the world, they don't have the C. Okay, the United States has the C, of course. So we have that in there because we don't want someone to say, well, it's following GMP, but they weren't following the right one. Okay, so I think these make sense to you. So cell culture, this is where we're going to grow up the cells. Okay, so this would be considered upstream. Okay, recovery and harvest is kind of an intermediate stage. This is where we're going to collect the cells or collect the product. Okay, and then we're going to purify. Okay, so we're going to separate the product from the cell debris. Okay, so all of this is considered downstream. Okay, recovery and harvest is kind of like an intermediate. Okay, so formulation means we're going to take the product and we're going to stabilize it and put it into a dose. Okay, so we're going to make sure that it's ready to go into the patient. Okay, and then fill means we're going to put it into the bottles, and then finish means it's going to be labeled and sealed up. Okay, sometimes the formulation and the fill and the finish can happen at another facility. Okay, so AstraZeneca is in Frederick. I think I spelled that wrong too. Is in Frederick. Okay, and they do some vaccine production. We'll talk about that more. And they do all of this down there for their products, but then this is sent to another facility to be done. Okay, and you would think that these two steps would be like easy and would be the last part, but it's actually kind of tricky. Okay, so here is a picture that I have. It's from the book, and it's a sample of the process flow, some of the equipment that's involved, and the time. Okay, so if we start here, okay, so they're starting up here. Okay, so I know it's kind of hard to read, but I just want you to get the idea. So inoculum. So here, what they're doing is they're taking the stock cultures, okay, and they're going to grow up the stock cultures and they're going to get it into a bigger volume so they have more cells. Okay, media prep, so they're making media, okay, and then we go into this step that's called fermentation. So what they're doing is they're taking the cells and they're expanding them and they're growing them up. So in this case, they're up to 25,000 liters. Okay, so when we get into the STEM building, I want you to look at our water collection chambers. They're on the 3rd floor. And right now, I can't remember how big they are, but it says on them. So we'll look at that and you can compare the size of that to this and see how much media it would actually take to grow these cells up. Okay, so here's 55 days to get enough cells and enough product to start harvesting. Okay, so then they're going to centrifuge and they're going to filter and they're going to try to collect the product. Okay, and then we're coming down here and all of this. So this is all upstream. Okay, and then we're downstream. So downstream, what we're going to start doing is we're going to start doing chromatography, which we'll talk about. Okay, and then they use chromatography columns, and there's going to be different kinds of columns, which we'll actually do most of these in lab. Okay, and then we end up with, they're saying about 8 days to purify it. Okay, and they're putting it in a bulk fill. So it's going to be in this big stainless steel vessel before they formulate and fill and finish it in order to get it to that step. Okay, so it'll be what, how many? So maybe 60, 70 days to get a product going. Okay, so just overview. Okay, now general unit operation. So we break these down, and the book we have has so much information in it. They didn't. There's just a lot of stuff in there. Okay, so general unit operation. So the first thing where the materials are going to come in is called raw material storage and handling. So a raw material is anything that's going to be used in the process. So if they're making media, okay, it could be the sugars, it could be stuff to make buffers. Okay, so it's the chemicals that are coming in. Okay, and they're going to come into the warehouse, and there's going to be an inventory control system. So we're going to know how much is in there, when we need to order more. We're going to know where it comes from. Okay, so we need to be able to trace the raw materials, and we need to be able to prove that the raw materials are what the company that we bought them from says they are. Okay, they don't use wooden pallets typically because things will grow in the wood. So they use plastic pallets that are going to be easy to disinfect. Okay, so the next thing that's going to happen is the raw materials are going to get weighed and they're going to be dispensed. Okay, so they're going to take the raw materials and they're going to put them into a cleaner container so that they can be moved into cleaner rooms. A really important part of this is media and buffer prep. Okay, so in order to grow the cells, you need microbiological media, you need broth of some kind, okay, and you need buffers in order to purify it. Okay, these are typically the way that it works. So if this is a facility, it has many floors. And on the top floor, this is where the media and the buffer prep are. And then they're going to have everything connected with pipes. So you're not hauling media through in bottles unless it's a small amount. You're going to open up a valve to a pipe and then it's going to flow down. So they do things on the top so that gravity will pull it through. They're going to be centrally located. There's two terms here. So the piping and the vessels that these are made in, we have to be able to clean and disinfect it or sterilize it. So two procedures that you need to know are CIP is clean in place. And SIP is steam in place. Okay, so they have a way developed, okay, that you can clean these big, huge vessels and the piping without taking them apart. Okay, so you can clean it and they verified that this process works. And then you can basically autoclave it or steam it in place. Okay, and then there's the lines are going to be called transfer lines. Okay, so another part of this is inoculum prep and cell culture. Okay, so there's going to be a biological safety cabinet in there because you want this to be sterile. You don't want to start out with your little tube of cells being contaminated. Okay, we have to talk about the classes of the rooms. So a class 100 room is pretty much a clean room, a sterile room. Lots of times they don't need a whole class 100 room. They just have a cabinet. Okay, so then there's recovery and harvest. Okay, so these rooms are where we're going to collect the cells and the product. Okay, so they have homogenizers. So homogenizer is kind of like a blender. Okay, they have centrifuges, which will spin the cells down. Most of these systems are closed because we don't want anything to be contaminated. Okay, if the vessels are big, everything's going to be cleaned in place. Um, Purification is downstream. Okay, so everything has to be a certain temperature. So if the product, so if you think about this COVID vaccine, right, and the mRNA needs cold temperatures. Okay, all the work is going to be done in a cold room, right? So the room is going to be cold. You have to wear your gloves and your mittens. Okay, the chromatography columns, it'll be easier for me to show you this when we get back to lab, but we use tiny little columns. They're just, a couple milliliters big. The columns that are used for purifying vast amounts of product are going to be huge. Okay, so they put them on these things called skids. And I'll find a picture. I'll show you pictures of stuff, but they call it a skid, so they can be moved around. We'll talk about this kind of filtration. Don't just hang on to that for right now. Okay, so fill and finish is going to be done in a very, very controlled environment. We don't want to get to the very end and start filling the bottles and have it get messed up. It happens a lot. It's all going to be sterile. We don't want any contaminants. There's going to be filling machines and then lyophilizers. This is freeze drying. Okay, so we have things in little ampules, little glass vials, right? And it's all dry and fluffy in there. And then we'll add a buffer to it when they need to inject it into a patient. Okay, so the idea is we want a product that's safe, pure, effective, and high quality. So we want to reduce the risk of contamination. So people and materials are only flowing one direction. Everything is unidirectional. Okay, and I'll show you in class, and I have a picture coming up. But if you go in one door, you're going to leave another door. Okay, so you're not going back and forth because you don't want to contaminate anything. Okay, so now the classes of rooms. Okay, so clean rooms are aseptic processing areas. Okay, so they have high airflow and they have HEPA filters, so high efficiency particulate air filters. So they're going to remove particulates that are 0.3 micrometers or larger in size. So the idea is that we're going to create a laminar flow. Okay, all of the clean rooms are going to be very controlled. Okay, so there's airlocks, there's restricted entry, and we're going to have pressure differentials. And I have a picture I'll show you. This is a silly picture. I took a class one time and the guy was really, really into filters. And when I made this PowerPoint, I was putting lots of pictures of filters in it. But this doesn't really even show you anything. Okay, a HEPA filter, or it's like a pleated filter. It's got really small holes in it so that things get stuck in it and don't go through. Okay, now a clean room, the materials in there are going to be smooth and hard and easy to clean and disinfect. Okay, they're going to be resistant to rust. We don't want any oxidation. We don't want any particulates in there. We don't want any particles in the air. Okay, so the air classifications. Okay, and I meant to update this. Okay, so we'll talk about this in class, but there are 3 broad categories, and there's different language that they use for this as well. Okay, so class 100 has less than 100 particles per cubic foot of air. Okay, the particles are anything greater than 0.5. Okay, and an example of something that would be a class 100, if it's set up right, would be a laminar flow hood. Okay, when you go into class 100, you go through, you have to gown in, and then you go through a pretty serious training when they let you do that. Okay, class 10,000 is less than 10,000 particles per cubic foot, and then class 100,000 is less than 100,000. Okay. So positive pressure is going to keep the clean rooms free from airborne contaminants. So these little pluses I have on here represent how much positive pressure there is. Okay, so here's a hallway and a common area, and it's only got one plus. Okay, so we have two airlocks here. Okay, so people, I know, can go in this way, right? But the air is going to flow out. Okay, so the gowning room has two pluses. Okay, the equipment airlock, because we don't want the people and the equipment going through the same space. We don't want anything to be cross contaminated. Okay, the air is going to always flow from an area of higher pressure to an area of lower pressure. Okay, so when I open this door, the air is going to come out when I walk in there. So that's going to keep any particulates that are out in the hall from going into the gowning room. Okay, then there's an intermediate room. So the area outside the filling room has got three pluses. So its air is always going to flow in this way. So our germy stuff is not going into there. And then the room where we're actually filling the materials, okay, where we're making the drug, putting it in the bottles, is even higher pressure. Okay, so the idea is the air that's out here has a very low chance of actually getting into here. Okay, so here is an example of what an airlock is, and you guys probably understand this. So, and some banks have these. Okay, so the idea behind it is that you can't open two doors at the same time. Okay, so the barrier between the outside and the inside is you can't open both doors at the same time. I'm not explaining things very well today. Okay, so here's the hallway. Okay, then here is the gowning room, and then there's the personnel airlock. Okay, so I can go in here. I can't open this door until this door is closed. Okay, and this door won't open until this door is closed. Okay, and same thing here. Okay, and then there's same thing here. Okay, so you can't open both doors at the same time. I don't know why I put a whole page on here, but everything is very, you have to have a key card to access. It's pretty secure. Like when we go to tour someplace, I have to send them a list of who's coming to tour a couple weeks ahead of time. They're going to make sure that you're not going to try to steal their information. Okay, so I have another slide for this, I guess. Okay, so it's going to limit access to outsiders. It controls employees. It's going to allow us to have an emergency response plan. You're going to know where everybody is if something goes wrong. There are going to be security guards there. There's barriers, fences, alarms, and lights. Everything is pretty secure. Okay, now in the book, they go through the chapter and they describe each of these pieces of equipment and what they do. Okay, so I'm not going to read those to you. I think most of it is self-explanatory. Okay. Here's more. Okay, laminar flow hoods. We'll talk about this in class. So the idea behind the laminar flow hood is that the air is going to flow through the filter, okay, through these HEPA filters, and then when it comes into this space, it's going to be sterile. Okay, BSC is going to protect both filters, centrifuges. We'll go over all of this stuff. Okay, so there's a lot of instrumentation that's used. There are engineers or technicians that understand how it works and they can control these systems. So there are systems that are going to check, sense things. They're going to indicate if something's wrong. They're going to transmit it to someplace else, and they're going to control it. Okay, a lot of it now they try to make it automated, so somebody doesn't have to be sitting in there babysitting it all the time. The information will get sent to someone's maybe cell phone that they have on. Okay, so it's going to check things like temperature, okay, pH, maybe cell growth, okay, oxygen levels, carbon dioxide levels. Okay, all those things are what's going to be regulated. Okay, process utilities. What are all the things that have to go into this? So I met with the we used to meet with the vice president for the facilities at AstraZeneca in Frederick all the time. And one of their hugest expenses is water. They use a ton of water, right? And they use a ton of electricity. So those are things that have to be taken into consideration when someone goes to build a facility. Do they have the infrastructure there to support it? Okay, when water is used, okay, we actually make a certain kind of water. It's called water for injection, or WIFI is how they say it. So WFI is WIFI. Okay, so they take filtered and distilled drinkable water, okay, and they're going to measure it. So it has to have less than 20 colony forming units per 200 milliliters of water. Okay, there should be no bacterial endotoxins in it, no added substances, and no objectionable bacteria. And one of them I have on here is Pseudomonas aeruginosa. Okay, so WIFI. Okay, now how do they sustain this? Okay, so the housekeeping is really super important. Okay, and there's preventative maintenance going on all the time. So there is a rotation of how they sanitize the rooms. So they're going to use different virucides, bactericides, and sporicides in a rotation to make sure that they don't develop any resistance. Okay, everything is going to be documented. Okay, so we're going to have SOPs, which are pretty much the same as work instructions. Okay, SDSs, which I didn't change this. They call them SDSs now. So these are safety data sheets, process flow diagrams, and then these piping and instrumentation diagrams, so P and ID. So these are basically blueprints that show you how it's gonna all be laid out. The SOPs are how you're gonna do the work. Okay, and I guess that's it. I thought I had I used to have a piping picture in here. I'll post one so you guys can see it. Okay.